Good day, this is the Professor, CBMD PhD. It's today Saturday, February 18, 2023. It is 9:59 and 3 seconds, AM Eastern Standard Time, and today's research, gluten. So let's hop to it here. New discovery to bulk up, gluten-free fiber supplement, date February 15, 2023. Source, University of Adelaide. The scientists for the first time constructed a reference genome for the source of the popular fiber supplement, psyllium husk, which can boost supplies of the versatile plant-derived product. The researchers extracted and sequenced the deoxyribonucleic acid, DNA, from leaf tissue to construct a chromosome-level reference genome for plain Tango ovata and used ribonucleic acid, RNA, from other parts of the plant to predict the function of its genes. And this is a significant development because it will pave the way for improvements to the quality and quantity of psyllium crops. As DNA is a molecule that contains genetic information needed for the development and functioning of an organism, while RNA acts as a messenger, carrying instructions from DNA to build proteins. As psyllium has been used for food and medicinal purposes for thousands of years, the seeds of the plant are milled to produce a soluble fiber used in pharmaceuticals and supplements to improve gut health and control blood cholesterol. As psyllium is also a common ingredient in gluten-free food, the seeds in their husk are naturally gluten-free, and when mixed with water, produce a sticky substance that replicates some of the functions of gluten in bread. And this quality makes psyllium an essential ingredient in gluten-free bread, and it can be used in a whole range of other baked goods. With the market size of gluten-free foods expected to reach USD 8.3 billion in 2025, demand for psyllium is predicted to increase. The plant is highly susceptible to changes in environmental conditions and diseases, which not only affects the yield, but also the price and quality of this valuable commodity. As today, efforts to improve the quality and quantity of psyllium husk have been hampered by the lack of a reference genome. The development of a high-quality Tango ovata reference genome will not only help to boost breeding programs, but will also support laboratory-based experiments to better understand how carbohydrates in plants are constructed so we can tailor them for food and pharmaceutical usage. Researchers predict the availability of this reference genome will lead to the development of new cultivars with higher yields that are more adaptable to environmental conditions, and this will stabilize the production of psyllium products and seed or husk prices. It is expected that this discovery will accelerate further research into genetic improvement and breeding of psyllium. And this is the Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.